My hope is that this video will help explain some of the respiratory histology that you'll be looking at. Some components that we're going to be able to identify are the epiglottis, the trachea, and lung tissue. And when we look at the epiglottis and trachea, we are going to be looking at the different cartilage and epithelium that makes up each one of these and how we can use that to help distinguish what we're looking at. And then as we look at some lung tissue, we want to be able to identify a bronchiole and the alveoli. The slides I will be using come from the Ohio State Medical University Center, and here's a link that you can use. So here we are at the Ohio State Medical Center website, and I've clicked Human Histology, and I've clicked Systems Histology, and I've gone to the respiratory system. So the first thing we want to look at is the epiglottis. And so here's a relatively small portion of it, and so I'm going to magnify this a little bit, and we'll look at the cartilage first. It's right here in the center. And I can tell that this is cartilage because I can see the lacunae with some chondrocytes in the middle. Here's a lacunae, here is a lacuna that is dividing. And then all of these really dark fibers that we see in here kind of running lengthwise, these are the elastic fibers. And so this tells me that this is elastic cartilage for the epiglottis. And remember that the function of the epiglottis is to cover the glottis when you swallow so that food does not go into the trachea. And so we want it to be able to shift position and then be able to go back to its regular shape after you have done that. So then let's look at the epithelial tissue here at the outside. So this is the first layer of cells of this epithelium, and here's the outermost layer of cells. So I can tell that I have several layers, a couple different shapes in here, but the outer layer here is flat. So I have stratified squamous epithelium here. And if you want to be even more specific, this would be non-keratinized, because even these outer cells here are still living. So I have elastic cartilage on the inside, and I have stratified squamous epithelium on the outside, and this is the epiglottis. Now let's take a look at the trachea. So here, remember first of all that the trachea or the tracheal rings are in a C-shape structure, and then connecting the two ends of the C will be that trachealis muscle. So if I look at this cartilage here, again I can tell it's cartilage. I can see a lacuna with a chondrocyte in the middle, several of those throughout here, but there's none of that really dark elastic fibers running through this section, and so this is hyaline cartilage. And then right here I can see the epithelial tissue. This is a really good example here. This is the cilia on the outside. Kind of this dark line is the surface of the cell, and then the cilia are attached to the outside. And then this would be the basement membrane. And so it looks like there might be several different layers of cells here, but really they all are touching the basement membrane. And so this is pseudostratified, ciliated columnar epithelium. And this is a goblet cell, and this is a goblet cell, or sometimes called a mucus cell, helping produce the mucus that is required for the respiratory system. Okay, so just to recall, epiglottis had elastic cartilage and stratified squamous. The trachea had hyaline cartilage and pseudostratified ciliated columnar epithelium. Now let's take a look at some lung tissue. Okay, we want to be able to identify the difference between a bronchiole and the alveoli. Now the bronchiole, here's one right here. And as I zoom in here, we can actually see that it is still pseudostratified ciliated columnar epithelium, and there is actually a layer of smooth muscle tissue surrounding the outside of that epithelial tissue. And remember that we can have bronchodilation and bronchoconstriction based on what's going on with that smooth muscle tissue. And the reason that we have these bronchioles, these really small bronchioles, is to bring the air to the alveoli. And those are these little chambers here that we see in the lung tissue. And this is where the gas exchange actually takes place. And this would be lined by simple squamous epithelium, so very, very thin single layer cells. Technically they would be called type 1 pneumocytes, would be the simple squamous cells of these alveoli. And this is actually where the gas exchange takes place at these type 1 pneumocytes. So I hope that helps in identifying these different structures.